The aim of this procedure is to obtain quantitative information regarding microtubule plus N tracking protein, or plusTip dynamics, in living growth cones using the software package plusTipTracker. Here, we describe the steps for analyzing images of EB1 GFP dynamics in cultured Xenopus laevis growth cones, but a similar protocol can be used for other cell types. Following the acquisition of high-resolution live images of tagged EB1 in cultured embryonic growth cones, the first step in the procedure is to use the command plusTipGetTracks to detect the EB1 GFP comets, link the comets into tracks, and determine the parameters of microtubule dynamics. The second step is to use the command plusTipCTracks to visualize the microtubule tracks superimposed on the growth cone images. The final step is to use plusTip group analysis to analyze and compare the microtubule track parameters of groups of movies. The main advantage of this technique over existing methods, like using hand tracking to analyze individual plusTip tracks, is that this method allows for rapid and automated detection, tracking, visualization, and analysis. The first step in image analysis is to detect the EB1 GFP comets, link the comets into tracks, and determine the parameters of microtubule dynamics. This is obtained with the command plusTipGetTracks. Prior to image analysis, convert each time-lapse movie into a sequence of TIFF image files and save each series in its own folder called Images. Note that Images is case-sensitive. To begin analysis, open MATLAB application and type plusTipGetTracks into the command window. This will cause a new dialog box to appear. Click on Set up New Projects and select one or more of the previous TIFF image series by selecting the appropriate images folder. Upon